Hi, welcome back to another episode. So today we're going to discuss about sensory receptors. We'll get right to the topic. Now, one of the problems with this topic is that it's not given that well in many textbooks. It's given concisely, not enough for you to actually understand the gist of the topic. Okay, so I read multiple books, multiple articles as well, compiled all the information so that it becomes easier to understand and differentiate between the different receptors. That is where students end up having trouble. Okay, so we'll just go ahead with it. First thing is to understand what are sensory receptors. How do you define sensory receptors? So, sensory receptors, they are nothing but modified nerve endings. I hope that is clear. In your dorsal root ganglia, which is the first order neuron in any type of sensation that is happening in your body. Okay, so the sensations from your different parts of your body reach your spinal cord and go up. So in this whole process, the first order neuron, the first neuron that is there, that is your dorsal root ganglia. There's a neuron over there. And mind you, it's a pseudo unipolar neuron. So this neuron has impulses coming from different parts of the body. So a dendrite-like process is there which receives impulse and reaches your dorsal root ganglia, from where the next set of output goes to the spinal cord or medulla or wherever, where the next set of neuron is present. Okay, so this nerve ending of this dendrite-like process gets modified in the periphery. So that's what a sensory receptor is. And they are specialized and modified nerve endings. Okay, so they are specialized and modified nerve endings. And what do? They convert different forms of stimuli as in any sort of energy, into one that can be appreciated by your brain. Finally, the whole connection is moved through your nerves and finally reaches your brain. And your brain should be able to comprehend what exactly the stimulus was. For that, it should be converted into form your brain can understand, neurons can understand, which is your electrical signal. So what it does is, no matter what form of energy it is, whether it's mechanical, whether it is electromagnetic, okay, whether it is some sort of chemical, whatever, so, or even thermal for that matter. So it doesn't matter what type of form of energy it is, it ultimately converts it. So that is the role of the sensory receptor. So they are modified nerve endings, specialized and modified nerve endings, which convert different forms of energy into an electrical one. Is that done? No. Each of these receptors are specific for each form of stimuli. It's not like when you touch, you end up inducing pain and the person howls in pain. That should not happen. These should be specific as well. So that is why they undergo depolarization in response to a specific stimuli. So that is a definition of sensory receptors. So we'll just go to a recap of that. It's a specialized and modified nerve ending, which convert different form of energy into an electrical one in response to a specific stimulus. So there is something called sensory organ. So the sensory receptor, which is there, along with the non-neural cells that are there surrounding it, forms a sensory organ. Sometimes it's called a complex. Okay, so the sensory organ or the sensory complex includes your sensory receptors along with the non-neural cells that are there surrounding it. Okay, now we'll just take a brief look at this table over here. So we got mechanoreceptors. What do mechanoreceptors do? Some sort of mechanical stimuli, touch, pressure, right? Such, or you're moving and cells move in any way. So even when you hear pressure changes induces movement of the hair cells. So finally, some sort of movement, even your baroreceptors. So when there is expansion, volume expansion because of the blood pressure and the volume that is detected. So such mechanical activity are sensed by your mechanoreceptors. And then we got thermoreceptors. As the name suggests, whether it is warm or cold, it detects the changes in temperature. Then nociceptors are there. So nociception is a cognitive element when it comes to pain. So when you talk about pain, the cognitive element of understanding pain, that is nociception. Okay, so that is pain. So these are nociceptors. And then there are electromagnetic receptors. Electromagnetic receptors in our body, uh, when you take a look at your rods and cones, when light comes out, it knocks out an electron from the whole spin, and that results in a whole chain of reaction that starts with the Wolff-Virchow cycle. Okay, so that ends up stimulating a set of cells, right? So that is your rods and cones. So then the glutamate pathway and all that, and finally you'll be able to see based on how it stimulates it. Okay, so that is your electromagnetic receptors in us. Now in birds and animals, certain animals and all, there are different types of electromagnetic receptors. It says that birds can fly in certain directions based on the gravitational pull, based on electromagnetic pull and all that. So they understand there are certain set of receptors in them. So that is another set of receptor. Then you see a chemoreceptor. So this detects any sort of chemical composition in the body. Taste, for example. Okay, so ultimately, whether it's salt, you're just measuring the chemical composition, sodium chloride. Okay, so salty, right? So like that, chemical composition of the substance is detected in taste, whether it is smell, or for example, your blood. In your blood, the pH of the blood, chemical composition, PO2, PCO2, all that is detected. That again is a chemoreceptor. Okay, apart from that, your brain also needs to understand certain other things about how much osmolarity is there in your body, in your blood, how much glucose is there, and based on that, it should adjust many things, hormones, your functions like behavioral modifications, like how much you eat, whether you should stop eating, whether you should stop drinking, whether you should drink more, whether you should feel thirsty, and all that. So all these are regulated by these receptors. Okay, so there is taste, there is smell. Carotid body is for osmolality, and carotid, the baroreceptors which are there in the carotid sinus, they are actually mechanoreceptors because they detect changes in volume pressure. Okay, then blood glucose and osmolality of the blood, these are there where there is a gap in your blood-brain barrier. So these neurons in your brain directly sense all these things. Okay, circumventricular organs. Okay, so in this one thing over here, mechanoreceptors, this is where as I had mentioned earlier, students generally find it difficult to actually differentiate between how different these are. Okay, so that is where I'm going to focus on today. Okay, so the primary objective of today's lecture is to make you understand how different these things are. Okay, and then we'll briefly discuss about this. Okay, so and we'll wrap it up with that. Maybe later if you want it, properties of sensory receptors, and how they start acting, receptor potential and all that. I can discuss about that if you want later. Okay, so this is the first thing, a corpuscle. We got Pacinian corpuscle, we got Meissners corpuscle. Okay, so these corpuscles, just remember, are rapidly attacked. Whether it is Meissners corpuscle or Pacinian corpuscle, corpuscles are generally rapidly adapting. We'll come to understand it later why that is so. Okay, on the other hand, expanded tip receptors, Ruffinis ending, these are slow adapting. So one example of expanded tip receptor is your Merkels disc. Okay, so we got a Pacinian corpuscle. Okay, so generally you can see that there's a corpuscle outside like this, corpuscle covering it, encapsulating it. Okay, over here, this is your Merkels disc, which is the expanded tip receptor, and we got Ruffinis ending. So these are slow adapting and these are rapidly adapting. Please remember that. Okay, so students get confused again with this because questions come interchanging them in a lot of MCQs, and even when you ask students, they find it difficult to remember the difference. So each small difference that I told you will build upon it slowly and try to understand it. Okay, so Pacinian corpuscle and Meissners corpuscle are rapid adapting, while Ruffinis ending expanded tip receptor are slow adapting. Okay, now this Pacinian corpuscle and the Ruffinis ending, these two are lying deeper. When you take a look at your skin inside the epidermis dermis, this Pacinian corpuscle and Ruffinis ending are there deeper. Okay, and your Measness corpuscle expanded tip receptors are more superficial. So finally, what it means is that these things that are there deeper have a larger receptive field, while those that are there towards the periphery, right, they have a smaller receptive field. Okay, so okay, now apart from this two corpuscle, there's another corpuscle which is called a cross corpuscle. So this is with regards to temperature regulation. So this Ruffinis ending is also involved with your temperature. Okay, cross corpuscle also involved with temperature. Okay, so Pacinian corpuscle, Meissners corpuscle, this cross corpuscle. Okay, and we have Ruffinis ending expanded tip receptor. Okay, so along with this whole thing, just remember there are some hair cells which are present. So when you talk about hair cells, there are generally hair cells inside your ear, vestibular organ also. So they move in response to changes in pressure or movement or whatever, autoconia shifting and all that. Okay, so that stimulates because of mechanical movement. Apart from that, on your hair, sorry, in your skin, there are hairy areas of your skin. So your skin can be divided into two areas, one that is hairy, one that is not hairy. Glabrous skin is what we call your non-hairy areas of the skin. Non-glabrous are your hairy areas of the skin. So in your hairy areas of the skin, there are hair cells that are there with which has receptor activity. Okay, so there are nerve terminals arising from here. They form a lattice network over here. So we'll come to that. Okay, so to come back again, there's a Pacinian corpuscle, there's a Meissners corpuscle. Okay, we got Ruffinis ending, we got expanded tip receptors. So these are in regard to touch mostly. Okay, so along with that, I just discussed about something else, the cross corpuscle which is there. There's the hair cells also that are there. Okay, so we'll just come into more focus now. You already got to know that your Meissners corpuscle and Pacinian corpuscle, they are rapidly adapting, while Merkel cells and Ruffinis ending are slow adapting. And when we talked about the receptive field, we saw that your Meissners corpuscle and Merkel cells, which are there more superficially, have a smaller receptive field, while Pacinian corpuscle and Ruffinis endings have a larger receptive field like this. Can you notice the difference? So what it means is this Pacinian corpuscle, which is there lying deeper, okay, so that can detect a larger area of movement. So one receptor detects a larger field. Okay, so if I touch here or here or here, it is one deep Pacinian corpuscle that is going to detect all that sensation. On the other hand, when you talk about Meissners corpuscle or Merkel disc, say if I touch over here and over here, so the difference between them will be felt. It will be felt as being touched from two different points. That is because of your Merkel cells. Because each time you touch in these two different points, it is getting stimulated. It is actually stimulating two different Merkel cells. On the other hand, these 2 points, if you touch and with a sensation that's supposed to stimulate a Pacinian corpuscle, it's ending up stimulating the same Pacinian corpuscle at such a short distance. Okay, so that is what you have to understand over here. So in regard to the receptive field, the ones that are superficially, like the Meissners corpuscle and your Merkels disc, have a smaller receptive field. So if they have a smaller receptive field, localization will be better. So that is just what I explained right now to you. When the receptive field is smaller, the localization will be better. So whether you touch it here or here, the difference can be made out by your body. Yeah, your brain can actually sense the difference between where is being touched. On the other hand, if it is vague like this, the localization will be poorer. So superficial structures like this have a smaller receptive field and have a better localization, while those with a larger receptive field have a poorer localization. Now we'll come to understand what we had mentioned about your adapting nature. So the corpuscle, the Meisners corpuscle and Pacinian corpuscle, if you remember, are rapidly adapting in nature. Rapidly adapts. So here you can see a stimulus being given. Here also you can see a stimulus being given. So we've given a stimulus for the duration of that change when the stimulus is being given. Okay, so when that change was happening, your corpuscle, both the corpuscles ended up detecting it, and there is a spike train. Neuronal discharge happened. But after the indentation, if you continue pressing that indentation, what happened? You can see that the stimulus is still being given, but it has already adapted. It is already adapted, and after that there is no more spike train. So it detects rapid changes, and after that it adapts also. So these are good for detecting, small vibrations, sudden changes, and all that. On the other hand, if you give a constant stimulus, and to understand the nature of the stimulus and all that, okay, with a constant stimulus, you just press it, or I'm sitting here, I get the sensation on my butt that I'm sitting there. Okay, so that to happen, we need something that is slow adapting. Or while I'm sitting, that indentation will give a sensation in these rapid adapting ones. But when I continue sitting without much change, then that should be detected by your slow adapting. So that is why your Merkel cells and your Ruffinis ending come into picture. So you can see here there is a stimulus given, and you continue to give the stimulus. So so initially when there's a change, when there is a dynamic portion, during that time there is a stimulus, and that will result in spike train. And when a constant force is also given, there also you can see that there is a spike train because it has not adapted itself. So it takes time for it to adapt. Okay, so that is the difference between these cells that I've just mentioned. Okay, so we'll just come back to it again. Pacinian corpuscle and Meisners corpuscle. So if you remember, they are rapidly adapting. So this corpuscle, okay, this corpuscle that is covering it, it kind of acts like a filter. Okay, so slow, constant changes, slow, low velocity movements, or a constant force, that does not, end up making this neuronal spike train inside this nerve cell. Okay, so when you press it, at that time, the dynamic change will result in shift of the connective tissue fluid inside it, and will end up causing some sort of shear and stimulate the cell. But after that, there is no longer any change. So that, okay. So on the other hand, if there is a low velocity change, or it's like that, then it won't detect it. Okay, or it's a static component, it won't again detect it. Okay, so this corpuscle which is there outside it acts like a sort of a filter. Now we got the Merkels disc, expanded receptor, and Ruffinis ending, which are slow adapting, and we had already seen that they have two components. One is your early dynamic phase. So when there is a sudden change and your intonation displacement, at that time there is a dynamic phase, and later there is a static phase. So if you remember the diagram, you can well appreciate that it was kind of different. The spike train in the earlier dynamic phase was much more, but later spike train was lesser. Still, there were two components over there. It was still firing, okay, even though the frequency was a little different. Okay, so this is the next thing that you need to understand. Okay, then this also we mentioned just now. Pacinian corpus and Ruffinis ending, which are lying deeper, have larger receptive field. They have poorer localizing. I've already explained this, so I'm not going to further explain it again. And these have a smaller receptive field and a better localization. Okay, now we'll come to, with examples, try to understand this. You can see the skin over here. We have the epidermis, right? So over here you got the epidermal structure, and below this we got the dermis. So this is the epidermis, this is the dermis. The Merkels disc is in the basal layer of the epidermis. The epidermis. Okay, so epidermis has many layers. In the basal layer of the epidermis, you have your Merkels disc that forms the Merkels complex over there. Remember complex? Sensory organ with the non-neuronal tissue along with the sensory receptor. So it forms a Merkels complex over here. This is dermis, and you can see that dermis is abutting into it. So this epidermis and dermis are forming finger-like projections like this. So this dermal papillae that is there, that is where your Meissners corpuscle is situated. So this is your dermal papillae, and we got your Meissners corpuscle over there. In the dermis, you can appreciate the presence of Ruffinis organ and your Pacinian corpuscle. Okay, so Pacinian corpus is much more deeper, and you can see the presence of Ruffinis endings over here. So once you know the location of these structures, we'll go on to understand their functions and exactly where they act. Okay, so yes, so this expanded tip receptor, what does it do? So this is a Merkels cell is there from the Merkels complex. Okay, so this complex, I told you, includes the non-neural tissue as well. So they have a modified sort of epithelium, and then there is a Merkels cell which is involved. Okay, so when there is touch sensation happening on the Merkels complex, this epidermal structures which have been not epidermal, epithelial cells which have been modified. So these epithelial cells end up secreting neurotransmitters. So these seem to have synaptic vesicles present in them, which are now going to get released, and these release and stimulate your Merkels cell. So generally this synaptic vesicles and neurotransmitters are present in nerves and stuff like that. Okay, certain parts of your body in different areas you have some modified cells which can hold neurotransmitters in synaptic vesicles. Okay, so one example is here in your Merkels cell. So in this Merkels complex, with touch, what happens is they release your neurotransmitter, which is now going to stimulate the efferent terminal. Okay, this is actually slow adapting. We already established that. Okay, so this helps you understand the fine texture. So if you keep touching something, okay, and to understand what that object is, how much of curvature it is, what is the fine texture elements of it, okay, to understand all that is your expanded tip receptor, which is your Merkels disc. Okay, so if I were to take my phone, okay, and I hold it like this, so I have to understand where the volume controllers. I go there, touch it, okay, and once I hold it, I understand, okay, that is my volume control. I can feel the shape and all that. When I'm holding it, the texture of it, how hard it is, or what is the pattern that is over here when I touch it, okay, that is made out by your uh, Merkels disk again. Okay, so if I continue to keep holding it like this, I have the sensation from my stop button. So this sensation again, that is delineating these two structures and abutting into it, that static component that is there, okay, that is again by your Merkels disk. Now next up is your Measness corpuscle. We already established how it's in the dermal papillae. Now these corpuscles, they have Schwann cells which are present. So the outer part over here, there are Schwann cells that are there, and inside them there are Schwann cell layers, and inside it there's some sort of connective tissue, glycosaminoglycans and stuff. Okay, and we have the coiled nerve terminals coming out of it. So this, what it does is, it actually detects, it's a rapid adapting. I don't have to explain again. Okay, so it is rapid adapting. So it detects sudden changes, dynamic changes much better when compared to your slow adapting ones. Okay, but they don't have a static component. So a sudden change or vibrations are picked up. So both of these pick up vibrations, but this vibration compared to this is much more slower. That's all. So they're more superficially. Okay, so I'm holding it over here. I'm holding my phone in my hand. So while I'm holding it, say it started slipping down like this. So in response to that, my body should appreciate that it's slipping. Okay, so sudden dynamic change over there where it's slipping. So in response to that, what I'm going to do is I'm going to hold, increase my grip so that it doesn't fall down. So such an effect is due to your Meissners corpuscle. A sudden dynamic change, more superficially, as it rubs and grazes across your skin, is detected by Meissners corpuscle. And even low frequency vibrations. Vibrations generally, one after the other, when it starts stimulating it, it needs a rapid adapting system to understand it and differentiate it from a constant touch. Okay, so that is where your Meissners corpuscle come into play. So a lower frequency like 5 to 100 hertz. This is just a range. Actually, the more average value will be much more narrow. Okay, so around 30 to 50 actually hertz. But when you widen it out, it comes to 5 to 100 hertz. Okay, so what it does is, again, how helpful it is, adjusting the grip, small movements, low frequency vibrations and all that. And this Meissners corpuscle is present in areas which are not hairy. That you have to remember. Meissners corpuscle are present in areas which are not hairy. So that is the glabrous skin. So in the glabrous skin, which is not hairy, that is where your Meissners corpuscles are present. Okay. So later, next slide, I'll discuss something else to help you understand that. Okay. Next, we'll go to your Pacinian corpus. Pacinian corpuscles are lying more deeper. They have poorer localization compared to your Meissners corpuscle, but they detect faster vibrations. So earlier in the neuronal spike train and the rapid adapting one, if you had paid attention to it, you would have realized that your Pacinian corpuscle actually is much more rapid adapting than your Meissners corpuscle. Okay, so if you want, we'll just go back to it. Okay, so here you can see over here that this, during a change, it can detect this long. On the other hand, here it starts adapting at this point itself. So this is much more rapid adapting. So it can detect much more faster vibration. Okay, so frequency ranges much higher, 200 to 300. Okay, so this can detect much more higher frequency vibrations, so much more faster vibrations. Okay, so in the structure, this is very important. Pacinian corpuscle is very large compared to all the other receptors, much more larger compared to all the other receptors. So a lot of the research that has been done on sensory receptors has been done on your Pacinian corpuscle, because it's much more easy to elucidate stuff on this. So receptor potential and all that has been studied in Pacinian corpuses. Okay, so the structure of Pacinian corpuscle, you have two hemilamellae like this. Two hemilamellae like this. Okay, so in the outermost layer and a inner layer is there. So outer layers, there are around 40 to 80 layers of lamella Schwann cells. Not outer, sorry. If this is in the terminal, there are around 40 to 80 layers of Schwann cells, and the outermost layer, they will have perineural epithelial cells, around 30 layers. So two hemilamellar structures, okay, and they have 40 to 80 layers of Schwann cells, and outside some 30 layers of perineural epithelial cells are there. So that is the structure of it. Okay, and when you press it, it ends up forming indentation that is in movement, and of course connective tissue and some glycans and fluids are there present inside. Okay, so apart from this, I've seen some articles which have doubts query. We're not really sure, but some textbooks, some places mention that this could even be involved in some sort of fine texture understanding. Not only this, but even maybe Pacinian corpus can be involved in understanding fine texture. So I'll just keep it out there for you. Okay, we're still not sure about it. Okay, so that is it. All right. So another thing is, if maybe it could be like this, like a fast vibration is there. You're moving your hand across something to understand the texture. So maybe that is how this is helping it. It understands fast vibrations, sudden dynamic change. Okay. So when there is a, for example, in my phone, if I have to push on the silent button over here, that is very textured. So even if I have my eyes closed, I can move across it like this, and once I reach that area, I can understand the texture of it. Okay, so I have my fingerprints which are there, which when you rub across it will kind of amplify the vibrations, and maybe like that you understand the texture. So that is what I make sense of it. But again, uh, you know these there's not enough conclusive evidence, and methodological problems to how understand this also are there. So anyway, so apart from fast vibration, just know that it could possibly be involved in uh, fine texture understanding along with your Merkels disk. Okay, next is your Ruffinis ending. So we discuss all these are fairly uh, easier to understand. Now Ruffinis ending is kind of a little different. Ruffinis ending again are deeper, but what we realized is there are collagen fibers over here. Okay, so in the extracellular matrix there's collagen fiber, and inside the Ruffinis ending also there are collagen fibers. So they somehow are connected to each other, so that when there is a deformation in the connective tissue in the surrounding, that results in a deformation in the inside also, and that sort of activates your uh, afferent terminal. So such deformation is actually more with stretch apparently. So for example, when you hold skin and you stretch it, your skin gets stretched over here. Okay, so stretch seems to activate Ruffinis endings better than a skin indentation. Okay, so where can this be used? When there's a skin stretch, even when joint activity is a joint. Okay, uh, when you move fingers, fast and do webbing and all that thing, you can detect, and this kind of helps you not overdo it. So some sort of proprioception is held by your Ruffini ending. So along with your muscle spindle and Golgi tendon organ, this also helps you understand. Okay, so some sort of proprioceptive elements are there in this. Okay, and I just briefly mentioned that this Ruffinis ending is not only foot touch, but it can detect temperature as well. Okay, so that is your Ruffinis ending. So this is for a stretch. Okay, so skin stretch, joint activity, finger movement detection, proprioception, and finally your warmth. This is faster vibration, some sort of texture. This is lower frequency vibration, sudden movements. Okay, that are superficial and all that. And over here, they are involved with your shape, texture, curvature of objects and all that. So I hope you can understand now the difference between these, exact difference. Okay, so with one example of just holding a phone, you can actually discuss all these things. So I'm holding my phone, I'm not looking at it. Okay, I keep my hand here like this, I keep my hand, and when I keep my hand here and hold it, I can feel my stop button or the volume button over here. Okay, how much the curvature is, whether this is actually my phone. Suddenly if you give another phone to me, I'll feel that it's not my phone. Okay, so all that will be through your Merkels. Okay, to some degree texture can also be your Pacinian Corpus. So for example, I move across this button over here where there is a definite texture, and I rub across it, I can feel the texture. Okay, so that again could be your Pacinian corpuscle. Now this starts going, slipping down, I adjust my grasp. Okay, that is your Measness corpuscle. Instead, I put it on vibration mode, I keep the vibration on, and somebody calls me and it vibrates, starts vibrating faster. So this sort of vibration will be picked up from your Pacinian corpuscle. Okay, and then next is your stretch, muscle stretch, proprioception and all that, and that is your Ruffinis endings. Okay, next step, once these were discussed, we'll just come to your tactile hairs that are there. What exactly do these tactile hair do? So I told you Meisners corpuses are there in non-hairy part of the skin. So sudden movements, okay, or superficial sudden movements can be detected in Meisners corpus. So for example, if you close your eyes and all of a sudden in non-hairy parts of your skin, an ant starts moving. So that will be detected by a Meisners corpuscle. But on the other hand, in areas which are hairy, where Meisners corpuscle are there, such movements are detected by these tactile hairs. Okay, so they are not encapsulated like this. Okay, there is hair over here, they're not encapsulated. Okay, they have efferent terminals which are coming along the base, okay, and parallel to the shaft, and they form a lattice like network pattern over here. So they are just like this, fast adapting but not encapsulated. They're fast adapting and they detect movement in these areas. So in non-hairy areas, glabrous skin, it is your Meissner's corpuscle. In your non-glabrous skin, which is hairy areas, it is your tactile hair that detect these movements. Okay, so that is the difference between them. Then again, there is the free nerve endings. There is Golgi tendon apparatus, muscle spindle, Ruffinis ending. So your Golgi tendon is there in your tendon to prevent overstretching. The muscle spindle is there that helps you understand your contraction and the stretch and all that inside the muscle. And your Ruffinis ending, already I told you, detects stretch. So these are all involved in your proprioception. Okay, the free nerve endings, they also detect touch to some extent, a more crude touch. Okay, so that is something you have to remember, and these are also involved in some sort of proprioception. Okay, so we have all these things which are involved in proprioception in your body. Okay, next is your temperature. Temperature, there is warm, there is cold. So your Ruffinis are for warm sensation. Okay, along with that, there is your transient receptor vanilloid channels. Okay, so TRPV3, TRPV4, along with the Ruffinis, detect warmth around 30 to 45 degrees Celsius, and they're carried by C fibers. Remember that C fibers are slow fibers, A, B, C. In your Erlanger classification, there's A, B, C. C is the slowest. Okay. Cold, on the other hand, around 10 to 38 degrees Celsius. Okay. We got cross corpuscle. Along with that, just like the transient receptor vanilloid channels that we had seen earlier, just like that, there's a menthol, TPP M8M for menthol. So that, along with cross, detects your cold. And it's also mentioned that TRPV4 has some overlapping activity. So here there is A delta as well, C fiber and A delta as well. So if someone were to ask you, give you an example where a person is made to touch both a hot object and a cold object at the same time in two different hands, which sensation among these will the person appreciate first? We got C fibers here, which is the slowest. We got C fibers here, which is the slowest. But cold to some extent is also carried by A delta. So A delta is much more faster than your C fiber. So which would you appreciate first? That is your cold sensation through this A delta. Okay. So we got C fibers, we got A delta C fibers for this. This is your Ruffinis and crosses your cold temperature. Okay. And I told you a range of here, 30 to 45 degree, 10 to 38 degree. So what happens when this temperature goes beyond it? This results in pain. It is actually detected by your free nerve endings. If you make it too hot, it should elucidate pain. Pain is an important reflex in your body. Okay, if it becomes too cold, you should remove your hand from it. If it's too hot, you should remove your hand from it. So these, beyond this normal range for these receptors, are detected by free nerve endings. So free nerve endings, Ruffinis ending, cross corpuscle, along with your transient receptor and light channels and menthol channels, right? So they're all transient channels. So they detect your temperature. So let's just come back to this again. To recap, we have your mechanoreceptors. We went through the whole list of it. Okay, we understood the various tactile sensibilities, the Meissners corpuscle, the Merkels, Ruffinis ending, cross corpuscle. Okay, so compared to earlier, I hope right now when you look at this, you have a better idea of exactly what they're doing. Okay, and the free nerve endings, Pacinian corpuscle, muscle, spindle, Golgi tendon, receptor, and then for equilibrium and hearing, your hassles. So now you can actually describe and differentiate all these things much better. And we got the thermoreceptors, the cold and the warmth. I just discussed it now. Nociception pain again, electromagnetic chemoreceptors. Okay, so hope this helps you out. And if you like the content, please do like it, subscribe it, and share it to people who might find this useful. Okay, so thank you and do good in life.